रिकॉर्डिंग कर देना शुभ।
हम्म।
रिकॉर्डिंग कर देना।
ठीक है, हम लोग आ चुके लाइव। स्टार्ट करते हैं सर। ठीक है। गुड मॉर्निंग, गुड आफ्टरनून एवरीवन। हैप्पी न्यू ईयर। Happy New Year to all our viewers, and those are also follow us and connected to Bioingene. Uh, we are very glad, and it's a great start. We think that this time, after a long time, I managed this webinar, and sir must be very busy. That's why we today our webinar is around 1 hour, not as 2 hours and that long. So we are today we try to focus a different angle of plant science. It's related to entrepreneurship, related to business from plant science. So it's a great opportunity for us to learn and know how these things are ongoing in India specifically. But yes, our audience is global, so we try to focus the others aspect also if it is possible. But yes, these more the rules and regulations will be covered on basis of the Indian background. So I request Shoma to please start today's webinar, introduce Sir, and introduce little bit about bioengineering if possible.
So, as you have heard, today's webinar is going to be short and sweet. And so, welcome everyone to Bioengine webinars. This is a new year, and welcome you all. Bioengine is a non-profit organization created to promote plant science research worldwide. This is an open platform from which plant scientists can present the current advancements of their research to the world. We hope future scientists can gain perspective and inspiration. Hearing the esteemed plant researchers as they talk about their scientific accomplishments and their views on building the future of plant sciences, let us dive deep into learning. We are thankful to the speakers and viewers of our Bioengine family for your interest in the webinar series and for your appreciation of our efforts. You can register for Bioengine webinars through our website bioengine.com or our social media pages. You are now live viewing this webinar and you can watch it again later on the Bioengine YouTube channel. So please remember to subscribe so that you don't miss out on any of our events. After attending today's talk, you can apply for a certificate of participation via the feedback link, which will be provided in the YouTube chat after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is encouraging entrepreneurs and safeguarding farmers using plant tissue culture technology. Our guest speaker today is Dr. Ashutosh Pandey, a very successful scientist and group leader at the BRIC-National Institute of Plant Genome Research, New Delhi. He joined NIPGR in 2017. He has been working in the field of plant metabolomics and metabolic engineering for more than a decade. He has published over 70 research papers in internationally reputed journals such as the Plant Cell, New Phytologist, the Plant Journal, Plant Physiology, Plant Biotechnology Journal, Journal of Experimental Botany, to name a few. He is the Fellow of the Alexander von Humboldt Foundation, Germany, Associate Fellow of Indian National Science Academy, New Delhi. He is the recipient of the INSA Medal for Young Scientists from Indian National Science Academy and the recipient of the Professor Sushil Kumar Innovation Award. He has served as a member of the Indian National Young Academy of Science and PTCA India. Dr. Pandey is currently coordinating the national program of the National Certification System for Tissue Culture Raised Plants as the principal investigator. This is a mandate activity of the Department of Biotechnology (DBT), Government of India, established under the Gazette of India Notification Seeds Act 1966. The goal of this national program is to facilitate the production and distribution of virus-free, high-quality tissue culture plants for the benefit of farmers across the country. Sir, we are honored to have you on the Bioengine platform. I'm sure our global audience is excited and waiting to hear from you. So we can start with your presentation now, sir.
Thank you. Just
Yes, sir. Visible.
yes. Now it's on slide so more.
No, right now everything is okay.
Yes, yes.
Lights.
Okay. So thank you, Dr. Majumdar, Soma, and the entire Biogen team for providing me the opportunity to talk on this important platform about the use of plant tissue culture that was considered as a very old technique and so on. So now I will share that how this technology is helping farmers to get better yield of the crops, and how this technique can be useful to get entrepreneurship opportunity in this field. So I will tell about a government of India's program. Before this, I will for the audience and especially students, I will tell what basis of the tissue culture, what are the tissue culture, and how this technique can be useful for the industry as well as farmers. So basically, tissue culture, the concept of the tissue culture is totipotency. So totipotency, if we talk about in a very simple language, so the capacity of a single cell to grow into complete plant. So here you may see that these are the single cells. These are the embryonic cell suspension of banana. I am giving a example of banana to so that the audience can understand in a better way. So that, but it is not restricted to only banana. It is also applicable in other crops as well. So here we have isolated protoplast from embryonic cell suspension culture of banana, and then we can generate it to develop thousands of and lakhs of plants. And these plants are genetically uniform. That is true to type. Here this is there is another way that industries are using the micro propagation of the crop. So here they use the banana suckers as a mother plant, and then they use it in the aseptic condition in the synthetic media, and then develop again millions of plants from a single mother plants. And these all plants, before supply to the farmers, it is being tested for virus indexing and genetic uniformity to the government labs that are recognized under our platform, that I will tell you in later slides. So the basically the importance of tissue culture is that here whatever plant the farmers are getting is true to type to their mother plants. There is no genetic variation among the plant. As well as these plants are clean plants, that are these clean plant means it is free from virus and pathogens. So if I see the plant tissue culture technology, so the process of producing plants from the tissue culture of the desired plant in an artificial nutrient medium under controlled environmental conditions. Here you see that the X plant, that may be leaf, root, or any part of the plants, and it is grown in the synthetic medium. And here in this artificial medium, the plants grow and produce number of plants. And as I tell that this tissue may any part of the tissue that can multiply. So this plant, it may be leaf, stem, root, suckers, or any other part for the regeneration of a complete plant. And the importance of this, the plant produced exactly similar. That is true to type. to the mother plants in all aspects. And here, in the aseptic condition, strict adherence and microclimatic conditions require and care during the hardening process. Here, after the plants is when rooting is done, the plants is transferred in the hardening chamber, that is primary hardening units, and then secondary hardening unit to acclimatize plant, because before this stage plants um, are being grown in the laboratory condition, that is controlled light and temperature conditions. So if we directly transfer these plants in the field, so the plants will not acclimatize, and it will be very high chance to die. So before exposing it in the direct field, it needs to be acclimatized within hardening unit. So first a few days we need to put it in the primary hardening unit, then transferred in the secondary hardening unit, then the industries can is selling these plants. So this slide I just tried to prepare to explain what I want to convey. So plant tissue culture industries utilizes this technology for the mass production of superior grade planting material for the desired crops. So in this way, tissue culture is helping and our nature, industries, farmers, and consumers. So here, this is the win-win situation for all the stakeholders. Mhm. Here, the benefit of tissue culture, that a single X plant can be multiplied into several thousands of plants in less than a year time, and this allows fast commercial propagation of the new cultivars as well. So here another important in case of endangered species. Suppose some varieties are getting endangered, and so government cannot allow those plants can be damaged. So here the best thing in tissue culture is that the X plant usually does not destroy the mother plant. And in this way, the rare and endangered plants can be cloned into thousands and millions of plants safely to further multiply and cultivate in the field, so that these species can be protected as well. And another important thing is that if once the cycle is established, means the protocol of the tissue culture is established within the law, so it can continuously supply a plant throughout the year without the limitation of the season. So normally when we grow a crop, there is a specific season and then grow, then we need to wait for another season. But this is not the case in case of tissue culture. So once the protocol is established, the companies can generate, or laboratories can generate these plants throughout the year. So here the advantages of the tissue culture is that the plants will be true to type of the mother plant, as well as since here the rooting is developed with the some hormones. So here the root system is very well developed for the plants. Here you may see that roots are developed so that it has a better um, position to absorb the nutrients from the soil. And these plants are disease free, pest free, free, and virus free. So the planting materials are clean. And another advantage of tissue culture is that plants, all the plants are uniform, so they grow in the similar way, and so that the produce can be harvested at the same time in all the crops. So here what are the requirements of the tissue culture? So here appropriate tissue should be selected. So first of all, we need to clear that what plant, what species we want to multiply. So depending upon the crop, we need to select the appropriate tissue to start. Then here you may see that this is the tissue and this is the synthetic medium. So the another one important thing is to establish a suitable growth medium. So basically, the media provides basically a carbon source as well as inorganic salt to supply the growth is required. So here it may be solid, semi-solid, or liquid phase. In case of cell suspension, liquid phase. In here, this is semi-solid medium. Another thing is that aseptic conditions are required for so that the microorganism cannot grow within the medium, otherwise it will be contaminated. So here I am just sharing the one of the pick of one of the NCSTCP recognized company that here you may see that the manpower is working and another one is observing. This is she's a supervisor supervising that whether all the persons are doing the right thing or not, and then on the laminar all the cutting and transfer of the explant to different medium is being done within the laminar airflow. So here I am showing you the complete cycle of banana from generation of the sucker. Then sucker can be shifted in the synthetic medium, and then multiplication, then rooting development, then primary hardening and secondary hardening. Finally, this hardened plant is provided to the farmers, and then the plants is shifted in the farmers field, and then they get more produce as compared to sucker grown plants. So here this is the complete cycle of banana. However, here once the companies select the mother plant, there is a specific criteria of selection of the plant. That plant should be healthy, and there are some other parameters. So once it will be kept in the selected the mother plants, then virus 100% virus indexing is required for the mother culture. So here we need to make sure that what the mother plant which we selected is free from viruses or not. If it is infected with the virus, then it will be discarded before moving it into the multiplication. Once it will be clear that yes, the explant that is selected is free from viruses, then it is shifted into a multiplication medium. Then up to 7 cycle multiplication medium the company can grow, and then it is shifted into the rooting medium. Here you see the root is developed, then it is transferred in the primary hardening unit to acclimatize these plants, and after this we shift it into the secondary hardening, and then again all the batches virus indexing is required. And after once the industries ensure that yes, the batches are free from viruses, only then they are allowed to sell these plants to farmers. And along with this, they also provide certificate of quality and certified labels to the farmers, so that farmers have trust that yes, the plants that we are purchasing is free from viruses. So now the question is why certification of NCSTCP, and why the government thought that it is a requirement of establishment of certification system. So here, as I in the very first slide, I said that tissue culture technology is considered as an old technology. And TCPF means TCPF is a short form of this is a tissue culture production facilities that produce that multiply the crops in large scale and selling to the farmers. So these tissue culture companies were also operating since 80s. This is not any new technique. However, despite its potential, the sector lacked standardization. So there were not any SOPs were available, and this the absence of uniform quality protocols also requires serious risk to crop health and farmers' income. And in some cases, suppose plants are virus infected, and once farmers are planting these materials in their field, so they will not get the produce. The plants will if we talk about banana, so banana has CMV viruses. The grand name, the variety is naturally susceptible to viruses, especially CMV, and apart from CMV, some other viruses are also infect these crops. So it spreads like wildfire. So if the it will be contaminated, so it will destroy the entire crop. So, considering this problem and resulting its substantial economic loss, Government of India established national certification system for culture response to reform the development of agriculture in India through plant tissue culture technology for the increased production and yield of the crops. So, if we see the as a whole NCSTCP, so NCSTCP is a unique quality management system established by Government of India by Gazette notification under the Seed Acts with the objectives of certifying the production and distribution of disease-free and quality planting materials to the growers. And this program is being implemented by the Department of Biotechnology since the year 2006. So this is the DPT has defined a well operational structure for functioning of the NCSTCP program. So there is an affiliate authority. So DPT itself acts as an affiliate authority. Then certifying agency of the tissue culture raised plant. So DPT is the certifying agency. Then to monitor the activity and to provide guidelines time to time, a very high level of apex and program monitoring and evaluation committee is is constituted to monitor the activities of NCSTCP. So NCSTCP works with the close coordination of NCSTCP management cell, ATLs, and referral centers. So the responsibility of is divided for all the components. Basically, the recognition and renewal and grievance readers come under the NCSTCP management cell, and this management cell also works with the close coordination with the referral center as well as ATLs. And then the tissue culture production facility, that is after recognition of under NCSTCP program, they can submit samples for testing to our recognized accredited test labs. And then ATL prepare the test report, and every 20-year sample we revalidate at the reference center. And then we issue the, after confirmation of the virus indexing and genetic fidelity test, and if found everything is in order, then the ATLs issue certificate of quality and as well as certified labels embedded with the QR code. So that, and this shows that the plants are virus free and genetically uniform. So if we talk about the major activities of NCSTCP, so it includes recognition of the tissue culture production facilities, recognition as well as renewal, as well as development of SOPs of the plants covered under NCSTCP. So there is a list of the crops that is already covered under NCSTCP. Apart from this, if we receive request from tissue culture production facility, any crop that has importance, economic importance, and that industry wants to do the multiplication for the so that they can sell it to the farmers. So we regularly update our SOP, and we can develop the SOP for so that the other crops can also come in our system. So apart from this, the certification of quality planting material, that we tell that how we certify the planting materials. And then during this certification process, we also identify some plants are mother plants and batches are virus infected. So in that case, we ensure that these plants should be disposed as per the guidelines of NCSTCP, so that these plants cannot infect other plants. Apart from it, we also regularly do the capacity building and training by providing training at our ATLs, as well as regularly organizing awareness program in different states to create awareness about this program and how this program is helping to the farmers as well as tissue culture industries. Apart from this, we also provide accreditation to the test laboratories where the plants is being tested from the tissue culture production facility. So now the question is, who can apply for the recognition of the tissue culture production facilities under this program? So here, any tissue culture production facilities, either it is in public sector, private sector, NGO, or any other firm is engaged in the production of tissue culture plant with a minimum production capacity of 0.5 million plants per year can apply. So then the commercial laboratory should be operational at the time of submission of application. Sometimes people think that before the start of the lab, we get the recognition, but this is not the case. So tissue culture companies uh, have to make sure that their facility is in operational at the time of submission of the application. So after and the entire process of this certification as is centralized, everything is online. There is no need to come in person to visit and submit the application. People can submit their application at any place through creating a register at our site, and then they can submit their application. After that, a team of experts visit to the facility to audit infrastructure of the company, stability standards of the company, then scientific working of the facility, and then other parameters of the guidelines available on our website of NCSTCP. So this is just a snapshot showing inspection of our experts to the tissue culture facility. And based on their observation of compliance of all the mandatory parameters of the expert team, recommend for their recognition of the facility. So if we talk about the mandatory parameters of the tissue culture production facilities for recognition and renewal, so the facilities should be broadly divided into 4 categories. One is non-sterile zone of the tissue culture facility. Another one is sterility and safety parameters of the tissue culture production facility. Then sterile zone, and then hardening zone of the tissue culture production facility. So this is the list of the mandatory requirement, which is also available on our website of the NCSTCP hosted at NIC. So if we talk about the non-sterile zone, so here is the essential equipments required for the functioning of the lab should be kept in this non-sterile zone, as well as media preparation room, autoclave for sterilization of the cultures, bottle wash area, as well as the storage room, media storage room, and bottle storage room. And there should be a pause box. Then, if we talk about the sterility and safety parameters of the tissue culture production facilities, then the facility must have hand and leg wash area. Then dress change cubicle should be there. Then air curtain and air shower should be there. And within the lab, there should be a fluorescent strip marking should be available. This is mandatory parameters. Then fire extinguisher should be available, and along with this, smoke detectors and emergency exit should be available. Along with this, to make sure that the facility has uninterrupted power supply, the generator provision should also be there. And then the layout of the laboratory should be designed in a way that there should not be any crisscross movement between sterile and non-sterile zones to avoid contamination. The sterile zone includes media storage room, inoculation room, growth room, as well as AHU. Then we talk about the hardening zone of the TCPF. So it includes primary hardening units, and it also it must have a humidifier, insect traps. Then here a secondary hardening unit, and it should be a double door entry. So these are some snapshots of the site visits conducted by the team NCSTCP. Now the question is why tissue culture production facilities should do so much things to fulfill the mandatory parameters. What are the benefits of this recognition of certification of NCSTCP? So NCSTCP has strengthened the tissue culture industry through robust certification, quality assurance, and regulatory support, thereby improving farmers' livelihood, generating employment, and contributing to national horticulture development. So NCS TCP certification enables tissue culture production facilities to assess the subsidy from National Horticulture Board under Mission for Integrated Development of Horticulture for establishing tissue culture laboratories. So certified tissue culture production facilities are authorized to sell the tissue culture raised plant material across the country and are eligible to participate in the government tenders, expanding their market access and business opportunities. So certified TCPFs can get the planting material tested at NCS TCP recognized accredited test labs at a very subsidized rates. So reducing compliance costs while ensuring quality and phytosanitary standards. So these are the some snapshots showing the adoption of NCSTCP certification is mandatory in their tender documents. So these are the some tenders of the different states where it is clearly mentioned that only those companies can participate that are recognized under NCSTCP. So I'm pleased to share with you all that currently we have 126 recognized tissue culture production facilities across the country. So in these states here, you see that the presence of NCSTCP recognized tissue culture production facilities by India. However, still, if we see about the Northeast region, J&K region, and Karnataka, Kerala, so, as well as Goa. So we need to encourage the tissue culture production facilities working in yellow highlighted states to get their certification under NCSTCP. So if we talk about the SOPs are available, so in 12 SOPs and guidelines are for the 12 crops are already available under NCSTCP. So if we talk about the 12 crops, so that is apple, banana, bamboo, black pepper, citrus, potato, sugarcane, vanilla, date palm, pineapple, pomegranate, and teak. So SOPs of these crops are available under NCSTCP. So these are the procedures and standard parameters for the tissue culture production facility. So is here at the after selection of the superior clones, it should be done the virus testing to ensure that the clones are virus free. And here they do the testing by DNA sampling for reference, and after this selection they multiply the plants, and before selling it to the farmers again, virus free, to ensure that plants are virus free, the every batch should be tested as per the assigned batch size criteria at our accredited test lab, and after surety that this batch is free from virus, only then the tissue culture production facilities can sell these plants to the farmers. So accreditation of the ATL, if we talk about, so the test laboratories are responsible for testing of the tissue culture material for virus diagnostic and genetic uniformity for the purpose of certification. And if we talk about the current structure, so currently we have 5 ATLs located at different places in the country. So one ATL is situated in Pune, Maharashtra, that is Vasantdada Sugar Institute. Another one is Anand Agriculture University, Anand in Gujarat, and then GKVK, University of Agricultural Science, Bangalore, and then Indian Institute of Sugarcane Research, IISR, Lucknow, then National Agri-Food Biotechnology Institute, Mohali, Nabi. So these 5 labs are engaged for the testing of the tissue culture planting material for virus indexing and genetic uniformity testing for our accredited test labs. So after testing, the ATLs prepare the test report based on the test conducted in conformity with the standard protocols and guidelines. And based on the test reports, ATL issue the certificate of quality. Here you see this is the certificate of quality. So once the ATLs test that these plants are virus proof, they issue the quality certificate along with the certification levels on behalf of tissue culture certification agency. So here the this certified levels of the tissue culture plants are provided with the QR code. So this QR code provides the end user to trace back the history of the plants where the tissue culture raised plants are derived. So here the complete information is available, so farmers can easily verify that which mother plants is being used to develop these plants, as well as what are the where which lab were involved in the testing of this. So these every information required at the farmer level is already available in this certified level, which can be um, tracked back by this QR code. So once the TCPF is recognized, they become eligible to get their plants tested at our ATLs. And our ATLs continuously testing the samples from the tissue culture production facilities, and test reports shared with the TCPFs in a time-bound manner. And then referral centers again validate the testing by retesting every 20th sample from each batch of hardened tissue culture plants. The certified batches of the tissue culture based plants are provided with the certified labels enabled with the barcode. And the labels contain all the essential information such as name and variety of the crop, details of the company where the plants were produced, batch number of the crop, and name of the ATLs where the plants was tested. So all this information is also printed on the label in the form of QR code to ensure the authenticity of the printed information. So if we talk about the testing and certification activity of our program, so here this more than 1.54 billion plants have been tested under NCS TCP since its inception. And since 2021, we have more than 4.2 million plants have been detected for the presence of viruses, and these plants were discarded. In the last 4 years, we have tested 754 million plants that are certified as virus-free planting material, and these plants have been sold to farmers. So we also have a criteria in case of plants found virus infected, so how it should be discarded. The report of the tissue culture plants not approved for quality certificates. And ATL immediately informs NCSTCP management cell and concerned tissue culture production facilities for the disposal of the infected plants. Then the disposal of the plants is being done in the presence of ATL and NMC staff. So here you see the virus-infected mother plants as well as tissue hardened plants are disposed of according to the standard procedures, which prevented the infected planting material from reaching to the farmer's field. So here you see that in case of virus infection, the plants are being collected, and we ensure that these plants should be discarded, so that here you see the plants are being dumped, then it is being sprayed with the herbicides, and then again it is being packed with the soil, you see. So to create awareness about this program, we also organize awareness program at different locations. So in the last year, we have organized awareness program in ISAR Pune, Maharashtra, Anand Agriculture University, Anand, Gujarat, as well as IBSD Imphal, Manipur. So we have organized awareness program with the collaboration with the different institute that we have mentioned. In Pune, we have organized the awareness program in collaboration with the ISAR. In Anand, we have organized this program in collaboration with the Anand Agriculture University. And in Manipur, we organized awareness program in collaboration with the IBSD. And our awareness programs engaged participants from diverse sectors, including tissue culture production facilities, progressive farmers, officials from the DBT, as well as the state governments, scientists, academicians, students, and local entrepreneurs. And our awareness program catches media attention, both print and electronic media. And this is the just snapshot of showing some of the awareness program highlighting the media coverage, and the program is also being covered regularly by Doordarshan. Here I am showing just snapshots. National Certification System for Tissue Culture Based Grants एक राष्ट्रीय स्तर की योजना છે જેનો મુખ્ય હેતુ ઉદ્યોગતા સંગઠન અને ટિશ્યુ કલ્ચર છે. કૃષિ યુનિવર્સિટી પ્રક્રિયા જણાવ્યું હતું કે અત્યાર સુધીમાં 500 મિલિયનથી વધુ છોડ પરીક્ષિત થયેલી છે, જે કેન્દ્ર પ્રયાસ કરી પાકોના ઉત્પાદકતા વધારવા These are just snapshots. So our we always try to our coverage should be in local language so that farmers can understand in their native language the importance of the tissue culture and this certification system. Apart from it, we also regularly organize the webinars in different crops by the experts, by inviting the academician as well as industry leaders to talk about the importance of the crop and how the tissue culture is benefiting tissue culture industry as well as farmers. So one of the most important thing in this that this tissue culture has not only provides benefit to farmers and tissue culture industries, but it has also socio-economic importance. So in this tissue culture production facilities, large number of women workforce are working, and the tissue culture in the production facilities, mostly they are established in the rural areas, and where they provide employment opportunities to large number of women. So if we talk about overall structure, so minimum 50% and 90%, even in some tissue culture production facilities, 100% women employees are working. So these are the showing some snapshots of Novel Biotech Bangalore and Kailas Biotech Private Limited in Kolhapur, where 100% staffs are working are women. So here, women's employees acts as a skilled operator, and this provides them a safe and dignified employment opportunity in the areas nearby their villages, and they also contribute to their ability to financially support their family. So this is another importance of the tissue culture. So if we talk about the economic importance, so since our overall testing is around 85% of the banana. So I am showing you the example of banana. So if we talk about the cultivation of banana and production, so India is the largest producer of banana. So quality planting material in banana has benefited the farmers by providing financial security and enhance their income by ensuring the virus-free planting material for good produce and genetic uniformity for uniform growth and harvest. So if we compare the production of sucker grown banana plants as well as tissue culture grown banana plants, so farmers income increased approximately 30% if they use tissue culture rich plants as compared to suckers. So in this way, the adoption of tissue culture provides more than 35% better yield. So and similarly, their income is increased. And as a result of this adoption of the plant tissue culture in banana, so banana export, if we talk about in the past decade, so banana export increased more than tenfold. And with this, I would like to stop here, and to the audience, I would like to share that next month we are organizing 47th annual meeting of Plant Tissue Culture Association India, along with the symposium and industry conclave. So here we have invited several industry leaders to come and show their success stories. So our event will have inaugural lectures by eminent scientists and industry leaders, the keynote talks, as well as industry pavilions, panel discussion with the scientist and industrialist, poster session, and young researcher A to B and B to B awards for best presentation. So if we see, for the first time, a plant tissue culture industry conclave is being organized alongside this year's annual meeting of the PTCA to augment industry academia interaction and to boost tissue culture based biomanufacturing in line with the Bio E3 policy. So, Government of India and Department of Biotechnology launched Bio E3 policy to get the entrepreneurs in biomanufacturing. So, with this, I thank all the visionaries, experts, and stakeholders who played a pivotal role in the conceptualization, development, and framework of NCSTCP. So, this is the current team of NCSTCP. We thank our ATLs and referral centers and our colleagues from NCSTCP management cells. And to run this program, the financial support is provided by the Department of Biotechnology Technology. I also thank our director, Dr. Devasish Chattopadhyay, from BRIC and IPGR nodal officer, Dr. Manoj Modi from Department of Biotechnology as a nodal officer of NCSTCP. We thank all our committee members of APEX, PMEC, and accreditation panel members, ATLs, reference centers, and we thank all the NCSTCP recognized production facilities for their faith and confidence in this certification system. Thank you.
Thank you,
So
sir.
now I would be happy to take questions if any.
Yes, it's a great, lots of informations, and people are also asking lots of questions. But the general questions, what I have found till now, that for potato and for banana, which will be the best explant, means which part can be used for the tissue culture regenerated plant for potato
Basically,
and bananas?
yes, both have different things. So, in case of banana, if we see about banana is grown through sucker. So banana, in case of banana, they use sucker, and in case of potato, they use to develop the mini tubers. In case of potato, so they use some hypocotyle regions to develop this mini tuber in hydroponics, and then they have a better root system so that they can harvest mini tubers from them, so that it should be a better quality. So all the chips that we consume is being developed by this hydroponic system. And in case of banana, this sucker is used as an explant.
And sir, people are asking for some suggestions for prevent the contaminations in tissue culture. So what they can put into the
So
medium
yes,
and how?
very, very important questions. And this question basically is more important if we talk about in context of industry. Otherwise, once the contamination comes, the industry lost several bottles and entire batches of the plant. So the guidelines that we were talking about, our certification system, that there should not be any crisscross movement. So first of all, it is important to have trained manpower, so
Hmm.
that they can work effectively without contaminating the cultures. And then your the sterile zone and non-sterile zone should not have any crisscross movement. Otherwise, if people will frequently move within sterile area to non-sterile zone, then again come in sterile area, so the contamination will come. So the guidelines, whatever we have suggested, is to avoid the contamination. So if their manpower is trained and they follow the guidelines, then I hope they can significantly reduce the contamination.
Okay. And yes, sir. And one one question is that as a career development, you already mentioned the startup and all. But being a student from botany, what is the prospect to develop career in plant tissue culture? As a, I think,
Yes,
any student.
yes. So in tissue culture, basically, there is a lot of opportunity. So there is a misconception that yes, this is a old technique, boring technique, so we cannot develop the career, but this is not the case. Now, whatever presentation I shared is entirely based on the tissue culture. So here, once the person has a good understanding of the tissue culture and has expertise in tissue culture, so there are different very good opportunity. One, they can establish their own tissue culture production facilities for the production of the commercially important crops to sell these plants, because this is a very huge demand of the tissue culture plants. Another one, that person can be a advisor in different companies. Another one, they can develop their laboratories for testing. So there are so many opportunities because here, whatever I am saying, 126 industries are running. So they all have require a trained manpower demand. So there is a huge demand of the expert manpower in this field.
Exactly. And similar on a question line to your that particular this answer, the one asks that can you guide student regarding job oriented short term course at
So
NIPGR providing the tissue culture based skills courses?
not only NIPGR, several institute regularly organize training program on tissue culture. ICAR institutes, even NIPGR also organize workshop on it, as well as CSIR labs organize regular. If you talk about ISBT Palampur recently organized training program on tissue culture. Similarly IARI also organize, NIPGR also organize. So many opportunities are available. So sometimes people also ask, so we always revert to the different participants that where they can get their training.
One question is related to that what the minimum amount of rupees is required to establish a PTCA lab? Hello, sir. Are you audible, sir?
Yes, but
Yeah.
I could not hear you. Can you please repeat
Oh,
the
yes.
question?
The question is that what is the minimum uh, uh, amount or money is required to establish a just lab for propagating tissue culture or
It
into the tissue culture
Yes, it depends upon different
scale?
factors. Yeah, so at what scale the person wants to establish the tissue culture and which crop they want to start. So if we talk about banana, and if the person has their own land, so I think 1 CR will be enough, and government also provides subsidy,
Hmm.
so that if anyone wants to establish the tissue culture facility, so they can get the subsidy from state governments as well as Ministry of Horticulture. They regularly provide subsidy. So I also said in one slide that the government provides subsidy.
Yes, and your
But if we talk about in general, so I think one CR will be is required to establish the lab and other infrastructure, so that they can easily run their facility.
so that and my one last one is my personal question.
Yes.
Then I give you 4 5 minutes more to Soma. That when you certify that whether it is the virus infected or not, is there any option to check which variety exactly this is? Means any barcoding of the plant species? Means if there is a
Yes,
lot of
yes.
okay, so the certifications come with to the name of the species also.
I just here. Yes,
Okay.
here clearly mention that which variety, what botanical name, or what common name, genus as well as species, both are mentioned in this level.
Okay. Okay.
So
So,
very clearly, it is very important. That's why we carry out the genetic uniformity testing.
exactly. Otherwise, the output is different. Maybe plant is sustainable, virus free, but the product will be different if it is not
Yes,
tested.
yes.
Okay.
So
So,
that's why, because here it is specifically mentioned that suppose if we talk about banana, so banana has so many germ plots in different geographical areas. So if
mm,
people are purchasing the plant, it is their right to know which variety and which species they are purchasing and they are planting.
mm. So any testing for GMOs related things or
So no, this is yes, GMO testing is already available at different platforms. But since this is GMO is
not?
something about not
Mm,
we cannot directly sell. So here I think it would better
not
not
required.
to confuse.
Mm,
Otherwise, the audience will get confused about GMO and non-GMO. So here, whatever we are talking about is non-GMO.
exactly.
And in our areas, there is no testing of any GMO and other
Mm,
things.
mm. Yeah.
Only certified planting material from the recognized labs only.
Mm, exactly. So, sir, is a great knowledge, and I think we also get something from your interview session also. Soma,
Yes,
there is
talking about banana cultivation, sir, banana generates a large amount of waste biomass. So, are there any biotechnological innovations to convert this waste into value-added products and create a new income stream for rural
Yes,
entrepreneurs?
this question is very close to my own research interest. So if we talk about banana, so 80% of the total biomass of banana is basically agricultural waste. If we talk about pseudo stem, um, banana peel, banana flower, so in some part people use it for different purposes, but actually it is not being utilized the way it should be.
Hmm.
And banana, if we talk about banana pseudo stem or banana peel, has a lot of natural plant products that are very costly, and it is highly very huge demand of these molecules in cosmetic and pharma industries. So this in our lab we are working on one project, and several other research groups are working on different aspects to utilize these value added develop value added products from this agricultural waste. Some places people use it as a organic manure, and some cases some industries utilizes to produce ethanol. But there a lot can be done using these agricultural waste. If we talk about phenolic molecules, so there are various valuable flavonoids are present
Hmm.
within banana peel and banana pseudo stem that can be utilized. Last month, we have organized an awareness program in Imphal, and there are several MSME units we are working to utilize this banana pseudo stem to develop some uh, uh, colors and some type of shirts and woolen shawls from this banana pseudo stem threads.
Yes, yes. Sir, are there any
before
challenge?
Soma,
Yes.
before going, sir, you can stop the sharing also, so we
Oh,
can show you.
yeah, yeah. I
Yes,
forgot. I forgot
yes,
that.
yes. Now, okay, okay,
Sir,
Soma, sir.
are there any challenges in translating the lab scale tissue culture and metabolic engineering research into commercial products or startups?
Yes, because if we talk about in the lab scale, so we work on the certain things that our purpose is different. And if we talk about industry, so first of all, it is important to know that there should be a cost calculation. So industry will not run in loss.
Hmm.
So here, the in case of a large scale protocol should be optimized in a way that the protocol should be economic and it is affordable. And if we talk about metabolic engineering, so again it comes in some type of genetic modifications. So before moving it in to the large scale and at industrial scale, there is some regulatory guidelines where we need to get the approval from the at institute level IBSC and then at RCGM. So without the approval of RCGM, we cannot move anything from the lab to industry or lab to land. So
Okay.
RCGM approval is required in case of any genetic modification is being done either for metabolic engineering or for any traits like some type of stress tolerance or anything. So in those cases, RCGM approval is required.
Okay. Sir, other than the NCS TCP recognized tissue culture facilities, are there any other ways that young entrepreneurs can collaborate with research institutions to develop tissue culture based solutions for agriculture and healthcare?
Yes, yes. The tissue culture companies can collaborate in any government or private labs as per their convenience. This is not compulsory that NCS TCP recognized labs can only do the collaboration. Any labs are free to develop the collaboration. Like if we talk about in case of potato or banana, so ICR Institute is located in Simla, that is CPRI, Central Potato Research Institute. Several labs are regularly collaborating to get their hydroponics technology. Similarly, in banana case, NRC Banana Tirchi is a ICR institute, and industries are also collaborating with these labs. Even testing is also being done at different labs apart from NCSTCP, and but the testing rate is high in those labs. Even those are government labs, still rates are here. NCSTCP's purpose is not to earn money. We need to ensure that plants should be farmers get maximum number of virus infected and genetically uniform plants. So our rates are highly subsidized.
Okay. Sir, in the context of food security and nutrition, how important is the development of value added crops enriched with specialized metabolites for India's future?
Yes, this if we talk about biofortified crops, that is enriched with the metabolite and biofortified crops. The future is on biofortified crops because the it provides basically antioxidants and other health beneficial effect that is directly linked to our some phytopharma, or we can say that these molecules are considered as interface of phytopharma and nutrition. So
Hmm.
these molecules are very important. But again, the problem is that once it is biofortified, then again we need to get the approval of RCGM before it translating to. So there is a long process. But the good thing is that government of India is clearly provided the guidelines of genetic modified crops in case of gene edited crops. Like if
Okay.
the crop is gene edited, so in the guidelines is very easy and it can be sold out and transfer technology. We can transfer technology to industry.
Okay. Sir, we're nearing the end of this webinar now, and one last question. What advice would you give to the students and early career researchers who want to work at the intersection of plant biotechnology and entrepreneurship and farmer welfare?
Yes, basically, it is first of all, uh, the students should identify a problem. At what problem they want to work, rather
Okay.
than to initially to solve the problem, they should clear that what problem they want to address. Accordingly, they should select the their field and then they work. Then I think then they will better solve their problems and they can excel in their field. So rather than first to solve the problem, it is important to know the problem, what are problems
Hmm.
we want to solve, and in which our expertise is aligned. Accordingly, if we will select, then most people what first get the opportunity and then see that yes, this is the problem. Now we will work to sort out this. But it my opinion is different. That first you should know that what problem you can solve. Accordingly, you select the field, then you can better work. So science is basically, if you will work on passion, then you will never get tired. You will every time you will enjoy as long as you will work.
Yes, sir. Thank you, sir, for this advice. And sir, wish you all the best for your upcoming. It's a huge responsibility. Your upcoming PTCA meeting is there, and the entrepreneurship and leaderships and the collaboration that you're doing. It's amazing, and wish you all the best. And
Thank
Shubho,
you so much.
Yes, and let's see, sir. We try to join, but best wishes, best
Thank you.
wishes for.
Thank you so much. Thank you so
Uh,
much. We will sometime we will organize awareness program at ILS Bhubaneswar. Then we'll meet.
yeah, sure, sure. We'll love to. And as we are working on plant tissue culture and plants, so yes, we love to do that type of
Sure.
awareness
Thank
program.
you. Thank you.
And thanks a lot, sir, for
Most
joining
welcome.
and give your time. And
was
best wishes.
really very enriching, enlightening, innovative,
Yes, yes.
beautiful presentation, sir, and
Already,
really required in this at
Thank
this
you.
time.
If even after this any question comes from audience, you
yes,
send.
I already
I
got.
will send the reply.
Yeah, about 300
Thank you.
people right now watching. About 142 likes have been given to
Thank you.
your webinar, and
Thank you
lots
so
of
much.
questions.
Thank you.
Thank
Sir.
you.
Thank you.
Thank you, sir. Bye bye. See
Bye
you on the
bye.
next time. Okay.
Thank you, sir. All the best.
And thanks to all the participants. Please uh, put your feedback, and I already shared the link, YouTube link in the live chat box. And don't email us for that link. This link is only for the live viewers, but the recording is available in our website. You can watch anytime and multiple times. So it was the beginning of this year, and we try to bring more and more. And but due to time limitations and all, we are now try to bring at least one in a month. But let's see how we can go in that way. And Soma, can you little bit elaborate the application process? Means how they
Certificate application.
certification, because let's give audience 4 5 minutes more, then I will close the program.
Okay. So, Bioengine webinars, as you know, are free for all, open to all. You can attend these live international webinars and also receive a certificate of participation. You can post it on your LinkedIn and share it on your social media pages. The eligibility for a Bioengine webinar certificate requires you to be a registered participant. Definitely, you have to register first, otherwise, you will not be eligible for the certificate. And along with your attendance at the webinar, how are we checking that? We have to fill up the feedback form for your attendance at the webinar live hosted on our YouTube channel. Your attendance is recorded during, like now, Shubho will close up the feedback form. So you hurry up and fill it up. The feedback link is provided in the YouTube chat after the webinar talk. If you're interested in receiving a certificate, then please fill the feedback form correctly and submit it. After a process of verification, your certificate will be ready and you will be intimated via email within 14 days of the webinar. So after 14 days, you can receive your certificate. And please collect the certificate from our website, bioengine.com/certificates. It is right at the top of our homepage. You cannot miss it. On the certificate web page, you can find the link that leads you to a list of registered participants who submitted the feedback form successfully. So if the feedback form submission was successful, you will find your name on the list of registered participants. And after you find your name and certificate number, there's a next to your name, you will find a certificate number. And then you can download your certificate from the shared Google Drive. The shared Google Drive will have all these certificates with these numbers, and you just get your number certificate and you can download it. And please note that you have to be a registered participant. Again, I'm saying this. Also note that Bioengine holds the right to not issue or keep your certificate on hold depending on how you have submitted the feedback form. So if it is not submitted properly or you have made mistakes in giving your email address, if it is wrongly put, then it will be a problem. In the case the form is not completely filled, the email address does not match that of your registration form, your form will be considered void. So if both the email addresses have to match. So carefully fill the feedback form and always use the same email address. So you can register for such enriching, enlightening webinars through our Bioingene website, bioingene.com, or our social media pages. You are live viewing our webinar and you can definitely watch it again later at your convenience on the Bioingene YouTube channel. So please remember to subscribe so that you don't miss out. Every time we put something up, you get a notification. You remember to subscribe, so you will be part of all of our events. And we are happy to have you as our audience. And we are doing this all for you, the future scientists. So, Shubho, so now we can wrap up today's
Yes.
webinar.
Yes. Now we are going to close the today's webinar. Certificate link has been given. Receiving the applications already. Many people are submitted, and we received many applications right now. So see you in the next time. We intimate through the email and website and our social media. So please join us for the next webinar, and it will take some time to prepare the certificate and all, but as soon as possible, I will try to give the certificate and intimate all the registered participant by email only. And as Soma mentioned, certificate need to be downloaded from our website. We share it our Google Drive. Bye, everyone.
See you next time.